Thank you again. So it's really an honor to present after the last two speakers. So I'm hopeful I'm going to be somewhat near as interesting as both of you were. So I don't think I necessarily have to tell this audience how important genetics is or how much the field has changed in the last 15 years. This is an image from the Human Genome Catalog, just demonstrating how much of an explosion of data that we have had in the last 15 years in human genetics. And this is really, I don't even think modernizing the field is the right word. I think it's really changed the field, and it's changed the field of genetics, but I also think it's changed really what we know about disease and biology and disease mechanisms. And of course, this has been important both to just understanding disease biology and disease mechanisms, but I would be remiss in not commenting on the fact that it's of course really important to think about how we use human genetics for drug discovery as well. And this is some recent data from Open Targets, where they have shown that the majority of approved drug targets in 2021 have some level of human genetics evidence. And of course, there's some excellent examples out there, both of using human genetics for drug discovery as well as for safety implications. I really love the BCL11A story, where this is a target for gene therapy, but the association for fetal hemoglobin switching was identified in GWAS. And so I think this has the potential to be the first approved drug that comes from GWAS. And of course, another example that's fairly well known in the pharmaceutical industry is DGAT1, and a number of companies had programs for DGAT1 for metabolic diseases, and quite a number of companies had challenges in the with the safety and tolerability of this, and it turns out that it's because loss of DGAT1 is associated with chronic diarrhea, which, as it turns out, was a side effect in the clinic. So, some really great opportunities both to think about what therapeutics we should pursue, as well as what therapeutics that we might want to avoid. There are lots of really great sources of genetic data, but of course, one of the most important resources for the field as a whole are large population-based biobanks. Using the UK Biobank, which is an example that we're all very, very familiar with, where they have collected a huge amount of genotype data and now phenotype data, and that has been able to be used very extensively across multiple types of disease areas for genetic discovery. Our use of things like the UK Biobank and other biobanks has really just been exploding in the last 5 to 10 years as this data has become available. But with this explosion of data, one thing I think has really become obvious, and people have been talking about this now for the last 5 or 6 years, but I really have noticed how much press this has gotten, or how much attention this has really gotten in the field, which is that we are really, really falling down in terms of any kind of diversity in our genetic data. And people hopefully recognize the plot here on the left-hand side. This is from a paper that Alicia Martin published in 2019. And a similar figure from the GWAS catalog that's demonstrates their most recent data. And about 80% of the data in the GWAS catalog right now is based on European participants, whereas people of European ancestry actually only make up about 16%. And if you look at the pie charts there, you can see there's underrepresentation of almost every other ancestral group in the world, and really, really severe underrepresentations of groups of African ancestry and some other ancestries. And this has a lot of different implications. The sort of major obvious one is that we are losing out on an opportunity for discovery here. So a lot of other ancestries, a lot of other non-European ancestries have a lot more heterozygous sites than European ancestries. So just right off of the bat, statistically, we are losing an opportunity for new genetic discoveries by having this underrepresentation in who we're collecting genetic data on. But I think it goes far, far beyond just our inability to do discovery. There are also, I think, implications for health disparities. So again, this is from Dr. Martin's paper from 2019. And for those of you who don't follow Alicia Martin, her work is absolutely fantastic. This is something that she's very, very passionate about. She's sort of one of the people that I follow a lot, and why, I think has brought a lot of awareness to this field. So she has taken, if you were to take our polygenic risk scores for common diseases, and this is based on UK Biobank data, their European population and try to apply that to the non-Europeans in their population, what you can see is that there is a huge loss of accuracy in those polygenic risk scores. And you can see the global loss on the left-hand side and then by phenotype on the right-hand side. And I've sort of seen the estimates before. If you were to think about using, say, for instance, for height, if you were to use the European polygenic risk scores and apply those to Africans, the actual difference in your predicted heights would be on the order of a few inches, which is really probably not at all even phenotypically accurate. And on the right-hand side, you can see that actually, for some traits, it's not a huge difference. We must have a lot more shared genetic architecture, but for other traits, like the things on the right, there's a huge, huge, huge amount of disparity between what you would, between using a European polygenic risk score on other populations. Even above and beyond that, the translatability, this lack of genetic data also can have implications both for drug discovery and therapeutic effectiveness. So this is a recent headline from a drug called Plavix, which is a blood thinner, and it turns out that the metabolism of Plavix is somewhat due to a CYP2A19 gene, but it turns out the particular mutations in CYP2A19 that affect the metabolizability of Plavix very, very much by race ethnicity. So they're quite common in people who are of Asian descent, but much less rare in other ancestral groups. And so it's really, so clopidogrel is actually very much less effective, or it can be less effective in people who are of East Asians. And this is something that really needed to be addressed upon the release of Plavix, so that healthcare providers knew that they needed to test people or to think about how to prescribe this drug more accurately. So I feel like I sort of start off here kind of painting a really grim picture, but what I've also noticed, and this is really true over the last, I'd say 10 or 12 months, is that every time I look at the literature or every time that I look at Twitter, you are really starting to see a lot of data come out from population-based biobanks in other countries, methods to do better with PRS scores in non-European populations. So I think this is a work in progress, and I want to talk a little bit about some of the positive things and some of the really cool new data that's coming out. So of course, we focus a lot on UK Biobank because it's a resource that we use all the time. There's also a population called the Biobank Japan, and that's about 200,000 people. And they have phenotype data across 42 diseases, and they've been kind enough to make their summary statistics available for people to use. And there's some really interesting things that have come out of Biobank Japan. So first, there are, at least for a handful of diseases, completely novel discoveries. And so what I'm showing on the left-hand side is a completely novel discovery for CAD for a gene called ATG16L2. And you can see that if you were to look in a European population, there's just absolutely no association there at all. It's because the variant is monomorphic. But even above and beyond the new discoveries that you can make in Biobank Japan, using data from Biobank Japan and other resources like UK Biobank for a lot of diseases, it really helped to narrow down the set of credible variants for any one given locus. And so that really helps with being able to confidently say, this is the gene that we think is associated with this disease. So I think having more diverse data is fantastic, both for discovery of new genes, but it's also fantastic for actually just interpretation of some of the data that we have already. There are similar examples out now from Africa. This is a couple of different biobanks from Africa. The left-hand side is a novel gene for type 2 diabetes found in an African-only cohort. The right-hand side is also from a different African cohort, and here what I'm showing is a novel association for SNPs in the thalassemia range that are associated with total bilirubin. So this is a new connection between thalassemia and potentially levels of bilirubin. But in addition to that, the other interesting thing that has come out of some of this, again, is not just the new discovery, but just even thinking about how different disease heritabilities are across different populations. And that's compared here. So this is one of the African biobanks with the heritability compared to Iceland and Sardinia. And what I think is really interesting here is that Some traits actually have higher heritability in an African population than in a European population, but others have a lower heritability. And you could speculate on all kinds of different reasons this may be true, but I think we can really learn a lot, not just about genetics, but about biology in general. And it points to a lot of things like history and demographics as well. And this is a similar example where, again, the genetic discovery is really cool in this paper, but I like really thinking about what we can learn about demographic history and culture. And so this is a paper that has come out of the Mexican Biobank Project, which is several thousand people based all across Mexico, divided up by region. And you can really take a look, if you look on the lower left-hand side, and be able to look at how ancestrally different, even different populations in different regions of the country in Mexico are. And you know, there's a lot you can sort of learn about. This population looks different because of the history, because of who populated the country and when. You know, so again, even above and beyond biology, I think this is one of the reasons that I became a geneticist, just because it's so fascinating to really think about how our genetics have, you know, how much our culture has influenced our genetics and vice versa. And finally, these were all very country-specific examples or different continent-specific examples, but I think we can also focus within our own countries on making sure that we're doing a better job of having genetic representation based upon our country. And so, of course, people are familiar with the All of Us research program, which is US-based, it's population-based, and they've made a really concerted effort to actually enroll populations who have been underrepresented in the field of genomics. And this is something I think that can be very, very challenging. And what I really admire, this is the first bit of data that's come out of All of Us, is that they are doing I think really a fantastic job here, and you know, succeeding at what they set out to do here, which is having a lot of representation from non-European populations. And also they're really seeking to make sure that they're getting representation in LGBTQ populations as well. So I want to spend the last couple of minutes thinking about, hopefully I've convinced everybody of the importance of collecting data from populations from all over the world. But I think the other thing that's really important to talk about is how we do this. So I think doing this in an equitable and ethical and inclusive manner is just as important as doing it. And I think of sort of two kind of maybe these are a little bit cheesy statements that, you know, a rising tide floats more boats, and that if you give a person a fish, they can eat for a day, but if you teach a person to fish, they can eat for a lifetime. And sort of really the way to talk about this is to talk about how this has not been done necessarily well in science. And this is a term I learned recently. It's called helicopter research or parachute research. This is something that is not just limited to genetics. I think it's probably something that has occurred in all kinds of life sciences. And it's generally the concept that a high-income researcher carries out research in a research-poor setting, a lower middle-income country, without involving, I think the important thing here is, without involving any local communities or researchers. We can think of helicopter research in a couple of different ways. It really can be people from a US or a Western European institution going into a lower middle income country, collecting samples and then leaving. But I think we can also think about it in the same way that we think about the All of Us research setting. It can also be people going into an indigenous or a minority culture within our own countries and collecting samples and leaving. And we think about this as being sort of a scientist resource thing, but I think something else to think about is infrastructure. And so it can also be that in lower middle income countries, there can be lack of access to appropriate infrastructure. We think of being able to download and use the UK Biobank or 1,000 genomes, but that's because we have the compute resources and the ability to store that data. So this can also be a disparity in infrastructure. So there are some guidelines, some ethical guidelines on how to conduct research, how to conduct research in low and middle income countries equitably. But a lot of those are, what can a single scientist do? What has worked for certain research collaborations? And there's a big push, I think, again, in all of life sciences to really think about how do we give these guidelines as a field so that funding institutions, journals, et cetera, can really think about this and drive systemic change. I suspect there are probably a number of examples of institutions that are trying to tackle this. I just wanted to highlight a couple of the ones that I found. One is this group called the Global Ethics Code, and I came across this looking at Nature Publishing's guidelines. They now ask that people follow this type of guideline. You self-certify now when you submit a paper, and it's codes of conduct developed by a couple dozen organizations, several countries, both not-for-profit and for-profit companies, and it contains 23 articles, and they cover things like fairness and respect and care and honesty in scientific research. The second one is something called the Cape Town Statement, and this is a conference that goes on every year in Cape Town, South Africa. Again, not just based on genetics, it's based on research ethics in general. But the Cape Town, this group is planning on releasing a statement which will provide guidelines to institutions on ethical and equitable collaborations. So all of that being said, while I do think that it's important that we think about these things as a field and not just about scientists, there is some great work out there from a couple of different genomics groups where they have talked about, this is our experience, this is how we have made things work. And I think there is a ton that we can and should learn from their experiences that can go into a systematic guideline. And again, both of these papers have a large author list, and Alicia Martin is, I think, an author or a co-author on each one of these. And so again, I encourage people to check out her work if you have not. And they really talk about what are the different things that have contributed to inequity in research. And I think one really important thing is that this is not a one-size-fits-all problem, and it's probably not a one-size-fits-all solution. So in some countries, it's that there's government instability. In other countries, it's that there's lack of infrastructure. There can be lack of capacity. There's lack of a workforce. But there are certain guidelines, I think, that we can follow that are broadly applicable to both of these, and they lay out here a roadmap in those different sorts of guidelines that you can follow to establish equity in research in any setting. And then there's a more specific example, and this is from the NeuroGap Psychosis Research Study and Ginger Training Program, which is a large research cohort set up to study psychiatric diseases in Africa. And this really is a true partnership between scientists here in the US as well as scientists in Africa, where they have set up, training programs in Africa, where they have made sure that, students have opportunities to learn, where faculty here go to teach, and they emphasize a number of things that have really helped this project to succeed. And the first is local collaborations are essential. So really the thing we want to avoid here is having experts go into another country, collect samples, and not have the local population and the local expertise get involved. So really having those local collaborations is really important. And talking about what those local collaborations look like, what are those expectations? That has to be laid out as the project is being developed. You have to think about what's an equitable practice for collecting samples, what's an equitable way to share samples, who gets to analyze the data, how do publications work? And I think funding, needs to be really flexible. This is, I think, a very different mindset than all of us are used to, where there needs to be more upfront funding. There needs to be some flexibility in funding because you don't anticipate things like the internet is not as stable. That's something that maybe needs to be built. Compute infrastructure needs to be built. And so you need to have the flexibility to really be able to fund those unanticipated costs. In-person meetings are really, really important. And I think we've probably all learned that is true over the last couple of years while we haven't been having them. But just really to be able to actually have that honest face-to-face collaboration and team building and ability to build trust. So that's really important. And then finally, capacity building is really, really critical, and that the best way to achieve your milestones is to really think about capacity building first, and then that makes it much, much easier to actually hit your research milestones. So again, I think this is an incredible field, the field of genomics, and I think we are sort of just at the stage now where we are starting to be able to build these types of collaborations and grow this type of research. And I think as a field, we really have not just the opportunity, but the obligation to expand our research beyond European populations. But I also think that how we undertake this is just as important as undertaking it. And with that, I am happy to take any questions.